Good afternoon. Appreciate the opportunity to tell you about what I think is an incredibly exciting technology and explain its relevance in the cell and gene therapy world to you. No forward-looking statements from us. We're a private company, so you're spared that. We're a synthetic biology company, and our whole mantra is all about putting the power of precise control over gene function in the hands of product developers. And I'm going to explain to you exactly how we do that and why it's important. So I really see this field of being able to regulate gene function as being the next key frontier that we as an industry face in cell and gene therapy. I think we've heard many examples today of the huge progress we're seeing in the march towards gene medicines and the cures that they're starting to generate, and the focus, in particularly for our technology, is in gene and cell therapy, also immuno-oncology, but also gene editing, which we haven't heard so much about today. But we're seeing huge progress. I think key areas of progress is obviously the genetic understanding of the basis of disease and understanding what we can do to replace or edit or change those genes to ameliorate the genetic hand that we're played. We've heard a lot about, it's all about, as Tim says, it's all about delivery. So capsid, a lot of licensing deals, a lot of effort, and a huge amount of progress that's been made from the late 90s onwards in design and construction of novel capsids and being able to get the gene actually into the cell. And I think Tim's exactly right. It's all about delivery until you've delivered it. And the next question then is, well, how do you then control the gene once it has been delivered and get precision over that control? And as we heard from Amit, I mean, a great start to the day hearing Amit talking about his clinical data and the work that he's done in haemophilia. But from my perspective, I'd say, well, there's still some outstanding issues and consequences of those issues that we as an industry still face. And so we still see variation in clinical efficacy. We see variation in data. It can't all be to do with the gene. It can't all be to do with the capsid. It can't all be to do with the patients. But we're seeing this variation in expression level and efficacy. So are we going to end up with non-responders or poor responders? Or are we going to start to end up with a subsection of people who are going to be relevant for gene therapy or gene medicines? There's a big question over whether this is a one-time dose for a lifetime treatment or whether we're going to have to address multiple doses, and we've heard that from other people today. So persistence of expression over the patient lifetime has its consequences. Not every gene needs to be expressed at the maximal strength that you can possibly express a gene at. That can lead to toxicity issues, whereas we've also heard that other genes need to be expressed at a high level so you can reduce the vector dose and overcome some of the immunogenicity issues. And so having the appropriate strength of expression for the therapeutic effect to address toxicity or subtherapeutic underexpression is still an issue that's out there. Targeting of cell or condition-specific expression, we've heard about doing that at the capsid level, which gets you so far, but still a lot of capsids still end up in the liver, irrespective of their tissue tropisms. And so you're still seeing concerns over off-target expression resulting in potential safety issues that still need to be addressed. And then there's giving more control over the nature of regulation, whether it's to be able to induce regulation, dial it up, dial it down, or insert a suicide switch to turn it off. These are still outstanding issues that are very much at the expression cassette level of the technology that, in my thesis, are left fundamentally or majoritarily unaddressed in today's classes of products. And that's where we then come in and the focus of the Synpromics technology. So what we understand now is that it's only about 1.5% of the whole of the human genome which is protein coding genes, just this small sliver here. So the question is, well, what's the rest of the genome doing? Well, most of the rest of the genome is involved in determining when the gene is active, where it's active, how long it's active for, what strength it's active. The whole control elements of the genome is wrapped up in about 98.5% of the genome. And what we're focused on is mining all of these regions, this other 98%, to understand the pathways and the networks of interrelated gene activity and function at a cellular level in individual cells, to mine that data to create totally novel synthetic promoters that can be used to start. Here, this is an old slide saying that basically you've got delivery, you've got the gene of interest, and then you've got these accessory sequences. What we're saying is those accessory sequences, to overcome this next, the next issues of a regulation of gene therapy, those should no longer be seen as just accessory sequences, but these will become critical components in the design of products. And that's what we're focused on, is actually putting the power and control in these accessory components. And we do that with I'd say it's a very clever system. It's a very neat system based on bioinformatic data, machine learning algorithms to create our synthetic promoters. So at the end of the day, we can start to look at promoters currently used in the industry, which are either viral or endogenous, that have they have efficacy, they have function, but they have a number of weaknesses or a number of areas that can't be addressed and need unique tools to address them. So we've heard about the importance of size to increase packaging capacity of the vector today. We've heard about mentioned about being tissue type specific, about being able to choose your expression level, about being disease or environmental specific. We've heard about the challenges around IP and patentability. So each synthetic sequence, because it's non-natural in its nature, is patentable. So we can generate a huge patent estate from our technology. And we can start to do things like looking at multi-gene cassette optimization, stable cassettes of expressing multiple genes without interference from a common promoter or instability due to homology within a common promoter. So the way we do that, this is it encapsulated in simple terms, but essentially we have a bioinformatics data input that contains elements of all of the gene control elements of the cell line that we then put through a multidimensional discovery engine that incorporates many levels of component, not just enhancer elements and transcription control elements, but other aspects that regulate the function of the cell. And then using that information, we then come out with this totally novel promoter sequence that is custom designed to a design algorithm of choice that's required to achieve efficacy or achieve ideal function. And we apply what is now standardly accepted as a synthetic biology principle of design, build, test, and learn. So our design phase, as I've said, we include these multiple different elements harvested from bioinformatics databases to design and build promoter constructs. That they can go into either a library construct or a rationally designed construct, and we synthesize and build these unique promoters. These libraries can be up to several million in terms of complexity and size, but we've developed the ability to actually individually uniquely barcode every promoter within one of those libraries, which would allow us to then do a screening or develop a screening methodology where we put the promoter into expression cassette of choice, expressing the transgene of choice into the animal model of choice, and then use next generation sequencing to identify the activity level of every promoter in that library and deconvolute that entire database that then goes into our big data-driven machine learning algorithm, which enables us to create proprietary data of our own, which can then feed back then into the next round of design, build, test, and learn. So we ultimately get an output of a promoter that's optimal and designed for its specific purpose. In terms of applications, then we're active in both bioprocessing, so we're looking at biologics production, but importantly, as a challenge within gene and cell therapy, it's vector manufacture to develop more scalable vector manufacturing systems based on stable cell lines. But in the cell and gene therapy product enabling, then these are the areas that we're focused in, and the analogy I'd use here is in each of these areas, we're essentially like the Intel chip within a computer. So we embed and code our promoter into other people's products. We're not developing our own products, but we're enabling other people's products through our promoters. So we've done a number of deals. So a quick history is last year we raised £2.1 million. Over the course of the next 12 months, we'd signed up and licensed out eight different programs with companies whose names you'll recognize here from the gene therapy world. That gave us a total deal value of over 100 million. We're revenue generating. We've generated revenue this year, as well as secured another about 2.2 million pounds worth of grants as well to support the growth of the business. So we're now growing rapidly. We're built up now from a team of about 7 or 8 people to now a team of 20 people based up in Edinburgh. So in terms of some specific applications, so in the gene therapy areas, we've been hearing today tissue condition or condition-specific promoters. That's what we enable. So you can get a certain amount of tissue tropism through the vector, but if you can back that up then by making sure your gene is only active in the target cell type of choice, that increases power. If the gene expression level is ramped up, it means the dose can come down, so that can start to address some of the concerns around immunogenicity. On the efficacy side, as I said, it's getting the expression level appropriate to the therapeutic effect. But it's also about designing promoters that aren't silenced in the way that CMV or some of the viral promoters are. So you can start to have reassurance over that lifetime treatment and expression over that lifetime treatment. And then when it comes to vector design, freeing up more space by using a smaller promoter to increase the packaging capacity to start to deliver larger genes is an important component here. And we're creating promoters around 2 to 250 base pairs. So we are talking significantly smaller than currently used promoters. In the gene editing space, so many of the issues here are similar to both cell and gene therapy, but when you're thinking about in vivo gene editing, many people are going to be using vectors to deliver the enzyme and guide RNA and have that expressed in the target cell type of choice. So again, having a tissue or cell type specific promoter that targets expression to the cell of choice is a critical benefit. Being able to control the duration of that gene editing event is also important, so you just don't get ongoing editing and re-editing and therefore increased off-target effect. And so being able to shut down the function at a point in time is important. And so we're also exploring application of our technology in the gene therapy, in the gene editing area. And then for cell therapy applications, so we've talked about sort of the single hit going after CD9, but we're starting to hear more and more of people exploring options around logic gates, Boolean logic, designing pathways into cells and having those pathways carried by the T cells. So if you can achieve that logic gate at the promoter level, that's hugely powerful. So we've shown that we can create inducible promoters. So a biological or an environmental effect can decide when a promoter and therefore gene activation is switched on and conversely also switched off. So as well as getting optimal expression level for efficacy, condition-specific expression for efficacy, they're, again, key attributes that we can deliver. So just briefly on the viral vector bioprocessing side, so we were awarded a £2 million Innovate UK grant at the beginning of this year to invest in developing this area. Most of you will be familiar that obviously scale-up of AAV is hugely challenging. We're restricted by triple transfection of often anchorage-dependent cell lines, and the scale-up beyond tens or a small hundreds of litres volume is hugely challenging, plus the issue that the majority of the vector actually produced doesn't carry the gene of interest, so the packaging efficiency is hugely suboptimal. And so we believe that by coming up with multi-expression cassettes that achieve the same stoichiometry of gene expression as you'd see in the natural virus, then we can start to improve both the efficiency of, or the scale of production, the amount of product per cell, as well as the packaging efficiency of that vector. The reason why people use transient transfection is that some of the genes, the helper genes, are toxic to the cell. So we've got the ability to put the expression of those toxic genes under an inducible promoter, so you can choose when in the production cycle you turn that gene on and have it expressed to then produce the product. So this is now a big focus for us to advance on the bioprocessing side and come up with the next generation of manufacturing platform for both AAV as well as lentivirus as well as retrovirus. So it wouldn't be complete with at least some data. So we heard about liver first thing this morning. So this is just an example of some candidate synthetic liver-specific promoters that we created here. Now Amit's not here now, I don't think, but we actually used Amit's LP1 promoter as the comparator here to see how our promoters compared in two liver cell lines and also in primary human hepatocytes. And this is in comparison to CMV here. And so you can actually see that we've got a range of liver candidates that are within about 2 to 250 base pairs in size that are anything between 10 to 40-fold higher activity than promoters currently used in the clinic. This is demonstrating specificity here. So here we did a counter screen against some of the production cell lines you'd use. One of the concerns is the expression of the transgene during production influencing productivity. I was waiting for the sign. Okay. And so you can see that we see no expression there. This is just quickly some animal data. This is a keratinocyte-specific promoters that we developed. This is showing CMV in a transdermal injection, showing expression in the muscle, in the skin, and across a whole series of organs. This is liver, as you'd expect. Whereas if you look at our keratinocyte-specific promoter, there's actually absolutely no expression in the liver or other off-target organs. And this is our board. So yeah, so hopefully I've convinced you that once you've delivered the payload, the next frontier is how that payload is controlled to enhance efficacy once you've got your gene into the nucleus or into the cell.